Hey everyone, and welcome. Today, we're going to crack open the Molecular Biologist Toolkit and take a look at one of the most important tools inside, the cloning vector. This is literally the tech that lets scientists copy and paste the code of life itself.
So, just imagine for a second that you're looking at the entire instruction manual for an organism, its genome. It's massive, and you want to find just one single sentence, one gene, and make a whole bunch of copies of it. How on earth would you do that? Well, that's the big question we're going to answer today.
And that brings us right to our first section, the gene copying problem. Because, it's one thing to find a gene, but it's a completely different challenge to make enough copies of it to do anything useful.
Yeah, a single copy of a gene is just way too small to work with. It's practically invisible. If you want to do something really important, like manufacture a protein like insulin or figure out what a gene actually does, you need to amplify it. You have to make millions, sometimes billions of perfect copies. And that whole process, that's called cloning.
But here's the catch. You can't just drop a gene into a test tube and hope it starts copying itself. It doesn't work that way. You need a special tool to make it happen. And as our source material says so clearly, without a cloning vector, this whole process is, and I quote, totally impossible.
So what is this absolutely essential tool? Well, the best way to think about it is as a molecular delivery truck. It's a little vehicle that we can load our gene into and then send it into a host cell that will do all the hard work of copying for us. So the official definition is right here. A cloning vector is a DNA molecule that acts as a vehicle to carry foreign genetic material into another cell. Its entire job is to smuggle our gene of interest into a host, usually a fast-growing bacterium like E. coli, and then basically trick that cell into making tons and tons of copies of it. And the undisputed workhorses for this job are plasmids. Now, if you look at this diagram, you can see these little circles of DNA floating around inside the bacterium. They're totally separate from the main chromosome. And here's the key part. They can replicate all by themselves. This makes them the perfect, tiny, self-copying delivery trucks.
So what exactly makes a plasmid such a great tool? Well, let's pop the hood and take a look at the essential components that scientists have engineered into a modern cloning vector.
Right, so this isn't just some random chunk of DNA we found. A modern vector, like the one you're seeing here, is a really sophisticated piece of molecular technology. We're going to break it down into its three most critical parts, which you can think of as its engine, its ID tag, and its cargo bay.
First up, the engine. This is technically called the origin of replication, or just ORI for short. It's a specific DNA sequence that basically screams, "Start copying here," to the host cell. As soon as the cell's own copying machinery sees the ORI, it gets to work and duplicates the entire plasmid, taking our gene right along for the ride. Okay, part two is the ID tag, or what scientists call a selectable marker. This is almost always a gene that makes the bacterium resistant to an antibiotic. Now you might be wondering, why do we need that? Well, it turns out that getting these plasmids into bacteria is a really inefficient process. This marker is our secret weapon for finding the few bacteria that actually accepted our delivery.
And this is just a brilliant illustration of how it works. After we try to get our vectors into the bacteria, we spread them all out on a petri dish that's been laced with an antibiotic. So any bacteria that didn't pick up our plasmid, well, they die. Only the ones that have our vector and its antibiotic resistance ID tag can survive and grow into these little colonies you see. It's a super effective way to sort the winners from the losers.
And finally, the third critical piece is the cargo bay. This is the multiple cloning site, or MCS. It's a short, custom-built stretch of DNA that's packed with a bunch of unique cut sites for different molecular scissors, what we call restriction enzymes. Having all these options makes it incredibly convenient for scientists to snip open the plasmid and paste their gene of interest right inside.
But of course, technology never stands still, right? And that's true for molecular biology too. Over the years, scientists have constantly been upgrading these vectors, making them smaller, way more efficient, and just a whole lot easier to work with.
Let's just take a look at how far they've come. On the left, we've got PBR 322, which was a total game changer back in 1977. And on the right, from just 6 years later, is PUC 18. Just by looking at them, you can tell the design is getting a lot cleaner, a lot more streamlined.
And when we look at the specs, the upgrades are obvious. The newer PUC vectors got way smaller, making them easier for the cell to copy. And speaking of copying, check out the copy number. A small tweak to the ORI, the engine, made it jump from about 20 copies per cell to over 500. That's huge. But the real genius upgrade was a totally new way to screen for successful clones. This new method is called blue-white screening, and it's just so clever. Here's the idea.
The cargo bay, that multiple cloning site, is stuck right in the middle of a gene called lacZ. If the lacZ gene is working, it makes an enzyme that turns the bacterial colony blue. But, and this is the brilliant part, when we successfully insert our gene into the MCS, we actually break the lacZ gene. The enzyme doesn't get made, and so the colony stays white.
And this image sums it all up. The process is called insertional inactivation. All of a sudden, finding the bacteria that had your gene of interest became incredibly simple. Just ignore the blue ones and pick the white ones. It was a massive leap forward.
Okay, so plasmids are amazing little delivery trucks. But what happens when your piece of DNA, your cargo, is just too big to fit? Well, then you need to call in a bigger truck. And that's where bacteriophages come into the picture. These are actually viruses that have evolved to infect bacteria. Scientists, being clever, figured out how to hijack them and turn them into cloning vectors. Since their natural job is to stuff a big viral genome into a bacterial cell, they are perfectly designed to carry much larger pieces of DNA cargo. To put that in perspective, a typical plasmid is like a pickup truck. It's great for hauling cargo up to about 10,000 base pairs, or 10 kb. But a bacteriophage vector, that's your semi-truck. It can easily handle cargo more than twice that size, sometimes up to 25,000 base pairs. Just goes to show you need the right tool for the right job.
So from these tiny engineered plasmids to repurposed viruses, these vectors give us this incredible power to isolate, copy, and manipulate the code of life. It is a toolkit that has completely changed medicine and biology. And it really leaves us with one heck of a question. Now that we have the toolkit to copy and paste life's blueprint, what should we build with it?